Unit 4, Lesson 2, The Chromosomal Basis of Inheritance. BioNovice video lecture. This lecture addresses the following 4 learning targets: defining key terms associated with genetics problems, deriving the proper gametes when working genetics problems, differentiating between gene and allele, and analyzing the inheritance patterns in pedigrees. Uncovering the chromosomal basis of inheritance began with Gregor Mendel in his study of pea plants. For example, Mendel bred true-bred pea plants. For example, these 2 plants here, the first one shown with purple flowers, the second one shown with white. When these 2 plants were mated, the first generation resulted in all purple-flowered plants, whether the plants were self-pollinated or cross-pollinated. In the final generation, Mendel observed a 3-to-1 ratio. For every 3 purple-flowered plants produced, 1 white-flowered plant was produced. Mendel reasoned that only the purple flower factor was affecting flower color in the F1 hybrids. Mendel called the purple flower color a dominant trait and the white flower color a recessive trait. The factor for white flowers was not diluted or destroyed because it reappeared in the F2 generation. Mendel observed the same pattern of inheritance in 6 other pea plant characters shown here. Each character was represented by 2 contrasting traits. The heritable factor that Mendel was observing is what we now call a gene. Mendel developed a hypothesis to explain the 3-to-1 inheritance pattern he observed in the F2 offspring. 4 related concepts make up this model. First, as shown here, alternative versions of a gene account for variations in inherited characteristics. For example, the gene for flower color in pea plants exists in 2 versions, one for purple flowers and the other for white flowers. These 2 alternative versions of a gene are now called alleles. Each gene resides at a specific location on a specific chromosome, as shown in this diagram. Second, for each character, an organism inherits 2 alleles, one from each parent. Mendel made this deduction without knowing about the role of chromosomes. The 2 alleles at a particular location may be identical, as in the true breeding plants of Mendel's parent generation. Alternatively, the 2 alleles at a location may differ, as they were in the F1 hybrids. Thirdly, if the 2 alleles at a locus differ, then one, we'll call it the dominant allele, determines the organism's appearance, and the other, the recessive allele, has no noticeable effect on appearance. In the flower color example, the F1 plants had purple flowers because the allele for that trait is dominant. Fourthly, now known as the law of segregation, the 2 alleles for heritable character separate or segregate during gamete formation and end up in different gametes. Thus, an egg or a sperm gets only one of the 2 alleles that are present in the organism. This segregation of alleles corresponds to the distribution of homologous chromosomes to different gametes in meiosis. Some additional useful genetic vocabulary that you should review include the terms homozygous, an organism with 2 identical alleles for a character; heterozygous, an organism that has 2 different alleles for a gene; phenotype, which is the organism's physical appearance; genotype, its genetic makeup or the types of alleles that the organism has for its genes; monohybrid, a cross between 2 organisms for the study of just a single trait; and dihybrid, a cross for 2 genes for particular traits in an organism. This diagram shows a dihybrid cross, and it is how Mendel identified his second law of inheritance. He did this by following 2 genes at the same time. By crossing 2 true breeding parents differing in 2 characters that are heterozygous for both characters or alleles, a cross between the F1 resulting dihybrids can determine whether the two characters or alleles are transmitted to offspring as a package or independently. Using the dihybrid cross, Mendel was able to show the law of independent assortment. The law of independent assortment states that each pair of alleles segregates or separates independently of each other pair of alleles during gamete formation. This law applies only to genes on different, non-homologous chromosomes, or those that are far apart on the same chromosome. Genes located near each other on the same chromosome tend to be inherited together. A modern tool useful for genetic analysis of inherited traits is what we call a pedigree. A pedigree is basically a family tree that describes the interrelationships of parents and children across generations. Inheritance patterns of particular traits can be traced and described using pedigrees. Pedigrees can also be used to make predictions about future offspring and can also be used in conjunction with probability laws to determine specific phenotypes. In the diagram here at the top is a key of what the different symbols mean on these pedigrees or family trees. Generally, the trait that is being studied in the pedigree is termed the affected trait. So an organism who has the trait that's being studied is affected, and their symbol on the pedigree is typically shaded in. Those individuals in the pedigree who do not have the trait being studied, their symbols are typically not shaded in. Occasionally, a pedigree will show symbols that are half shaded in, and those represent individuals that are termed carriers. They typically are affected by the trait, although they may not be affected by the trait. It depends on whether the trait is a dominant or recessive trait. And carriers are typically, in terms of genotype, heterozygous.